Yes. So, a few housekeeping things before we get started. The first one is that please make sure you write down your name if you want, because I will send it to the professors, so that they can check you for the extra credit points that got you to come here. So, but it's so good to have you here, and it is my honor and privilege to introduce Dr. Andrés Durantini to give us our talk today on advancing alternate therapies to eliminate pathogenic microorganisms. So, a little bit about Dr. Durantini is that he earned his bachelor's degree in chemistry from the National University of Rio
Yeah, that's
in Argentina in 2009. His passion for research led him to pursue a PhD in chemistry at the same institution, which he completed in 2013. Following his PhD, Dr. Durantini expanded his scientific expertise through a postdoctoral position at McGill University in Montreal, Canada, from 2014 to 2017. His dedication to research was further recognized in 2019 when he received a Fulbright Fellowship to conduct research in the Department of Chemistry at Brooklyn College. In 2022, he continued his academic contributions as a visiting professor at McGill University. Driven by his commitment to both research and teaching, Dr. Durantini joined SIUE, where he currently serves as an assistant professor. Throughout his career, he has authored more than 40 publications in peer-reviewed scientific journals, reflecting his dedication to advancing knowledge in chemistry and photobiology. Let's welcome him.
Hello, everyone. Good afternoon. Thank you very much for the introduction. So I know that my accent is terrible. So if you don't understand what I'm saying, just raise your hand and say it. Okay. So I come from SIE. Actually, I come from Argentina, but I work at SIE. So I just want to mention briefly. We have a nice master programs in the chemistry department. If you are thinking or go for a master's, then please remember this. So where is SIUE? So Southern Illinois University of Edwardsville is located about 20 miles northeast of St. Louis. Uh, so all the way on the other side, right there. And also we have the thesis-based shared science program is about 38 hours. We offer some graduate assistantships as well, and a lot of our graduate students get support from research grants. Okay, so here are some of the requirements if you would like to apply, and then we have two deadlines. One is now in April 1st, and then we have an early application for fall 2026. The deadline for that one is December 1st. As you can see, we do research in different topics. You can work on some synthetic organic chemistry, bioorganic, biochemistry, physical chemistry, inorganic chemistry, biophysics, and so on. Some of the faculties. If you would like to know more about them, you can just visit our website. And thank you very much. But aside from that, I'm here to talk about bacteria. So, and bacteria are by far the most abundant organism in our planet. We can find them everywhere in the bottom of the ocean, in our body, in our guts, right? And as you can see right here, um, we are more bacteria cells than human cells, but we need to worry that much because 90% of them are beneficial. Okay? So, for example, they help us to synthesize some certain vitamins. They help us to digest food. They are involved in our immune system, protecting us from a lot of external threats. But the remaining 1%, those are pathogenic, and these are the ones that you need to worry about. Okay, so as you can see, they are they're responsible to food poisoning, they produce a lot of hospital infections, nosocomial infections. There is also Lyme disease and a lot of other diseases as well. So in my lab, what we do is try to eliminate those pathogenic microorganisms. We develop different strategies, different therapies to kill those pathogenic microorganisms. So now when you get infected with one of these bugs, what do? You take antibiotics, right? However, the misuse and overuse of antibiotics for many years have made bacteria resistant to this type of treatment. Okay, they can mutate, they replicate very fast, so they can pass the genetic information that makes them resistant to antibiotics to the next generation or to their progeny in a couple of hours. Okay, so they mutate super fast. However, the misuse and overuse of antibiotics for many years have made them resistant to this type of treatment. And as you can see here, the map shows the percentage of isolates of, in this case, E. coli isolates that are resistant to antibiotics, right? So isolates that come from different infections. And in this case, for a particular E. coli strain, it doesn't mean that all E. coli are pathogenic, right? We have good E. coli, but some strains are very pathogenic. And as you can see here in the US, about 30 to 40% of these E. coli isolated from infections are pathogenic, right? So now going to the numbers, and well, this is now a couple of years old. As you can see here, this paper is from 2022, but the data that they were using from 2019, they show that the number of deaths attributed to antimicrobial resistance is at least 2 times higher, almost 2 times higher than HIV or breast cancer, right? So you can see here how important this is. And so besides a clear need for the development of new antibiotics, a lot of pharmaceutical companies drop the research and development of new drugs simply because they say that it's not profitable, right? Instead of working on designing synthesizing drugs to tackle these pathogenic microorganisms, they work on designing other type of drugs to treat cancer, chemotherapy, for example, right? Much more profitable. So, anyways, what it is known about antibiotics that they have multiple targets. So, for example, usually they can target the cell wall synthesis. We are talking about prokaryotic cells here, right? With bacterium, so they can target the cell wall synthesis, they can inhibit the protein synthesis, DNA replication, and these are usually the main targets. And as a secondary mechanism of action, they upregulate the tricarboxylic acid cycle, okay, the Krebs cycle. They increase bacterial respiration, upregulating Krebs cycle, and they generate a lot of, actually they increase the concentration of reactive oxygen, right? So some of the reactive oxygen species, as you can see, we have super oxide radical anion, hydroxyl, a hydroxyl radical, hydrogen peroxide, all of these are involved in what is known as penton. Anyways, so the reactive oxygen species are molecules that contain oxygen and they can react with different macros. Okay, they can oxidize the DNA, they can oxidize proteins, they can oxidize lipids that constitute the cell wall, right? And those kind of completely change the property of cell, right? So big amount of these oxidants, a lot of oxidation is gonna kill the cells at some point, okay? So now, extractive oxygen species are being generated in the cell all the time, either eukaryotic or prokaryotic cells. They are part of the aerobic metabolism of the cells, right? So they actually the cells need these oxidants, these radicals, because they are involved in many different metabolic pathways. Okay, so as you can see here, they help to maintain homeostasis or the normal growth and metabolism of the cell. However, when the levels of reactive oxygen species are too high, for example, in this case, especially when those reactive oxygen species are generated by exogenous external sources, such as UV, for example, here, some chemotherapeutics, and so on, that's going to generate cellular damage and is going to end up killing the cell death, or cell inactivation. That's how we like to call it. Inactivation is synonymous to cellular death. Okay? So, and this is the strategy that we use in my lab to kill these microbes, these pathogenic microbes. We use what is known as photodynamic inactivation. You will also find this as photodynamic therapy, depending if you want to use this methodology. If you want to use it to treat cancer, you're going to find it as photodynamic therapy. If you want to use it to kill microbes, you're going to find it as photodynamic inactivation or antimicrobial photodynamic therapy, as it shows right there. So we use photodynamic therapy to generate big amount, exacerbated amount, huge amount of reactive oxygen species that are reactive for that are cytotoxic for not only for bacteria but also for virus and fungi as well. Okay, so how does it work? Well, you need three components, three basic components. You need light, not a bulb, but light, a photosensitizer, and oxygen. So what is a photosensitizer? Well, that's a dye, just a molecule. You can have organic dyes, inorganic dyes. But the thing is that when you excite this dye with light in the presence of oxygen, because there, right? We have oxygen everywhere. It's going to generate this the reactive oxygen. Okay, but in a very big amount, so that the defense mechanism of this microbe can they can defend they can defend themselves against huge concentration of oxygen. Okay. In my group, we're also interested in phenotype. Okay. Actually, how phenotypic characteristic makes microbe resistant to treatments and also antibiotics and so on. Just a brief over here, genotype, what the genotype is, where we have all of our genetic information, 23 chromosomes in a human cell, one chromosome in a bacterium, right? And then, whatever that genetic information is going to be expressed and it's going to be transformed into proteins, and then that's going to define our phenotype, right? The physical characteristic that makes all of us different. And in the case of a bacterium here, well, the phenotype can be, for example, given by the composition of the pili, of the cell wall, the composition of the flagellum, for example, the composition of the cell wall, if they have more or less unsaturated versus polyunsaturated lipids and that type of things. Okay. Also, the number of membrane proteins and so on. Okay? So, in today's presentation, I want to share with you some results and some strategies for how we use these photoactive biocides or photosensitizers to study phenotypic resistance at a single cell level. Then I'm going to show you how we can we use them to fabricate different antimicrobial materials. And finally, we have a small connections with the environment. And I'm going to share with you some results about how we use them to study atmospheric oxidation processes. Okay. So, to study phenotypic resistance to reactive oxygen species, actually phenotypic resistance, well, it says to reactive oxygen species, but it can be to the photodynamic therapy, right, as well. That's what we use to generate those reactive oxygen. So we use this methodology over here, and I hope that you can this over this is refers to the fluorescence microscope. In my laboratory, we use a lot of different light microscopy techniques. dry field, dark field, and phase contrast, and of course fluorescence microscopy. So we make a chamber, okay? The chamber is a 3D printed cylinder. Then we glue that to a cover lip, okay? We have the chamber. And then in that chamber, we are going to add a bacterial suspension. And as you can see here, well, bacteria have flagella, they have villi. Villi is what they use to anchor to the surface. And then flagella is what they use to swim around, okay? So we add the bacterial suspension, we give them some time so that they can anchor to the surface, okay? Then we wash away the planktonic bacteria, the ones that are floating around, because otherwise we're not going to be able to study them or to monitor them using our microscope. They're just going to go out of focus, kind of diffuse, or swim away. And then we add a propyrium guide, that's a cell death marker. So the way that this cell viability marker works is that in the extracellular medium, it's going to stay off. And once it finds DNA inside the bacteria, it's going to be in the on state, which means it's going to become emissive. It's going to be able to emit fluorescence. Okay? So initially, no fluorescence inside bacteria with DNA becomes fluorescent. Okay? And then we add the photosensitizer. So we add the photosensitizer, we excite the photosensitizer using green light. Right, that green light goes through the objective, excites the photosensitizer. Photosensitizer generates reactive oxygen species. Those are going to oxidize the bacterial cell wall, are going to damage the bacterial cell wall, and that's going to allow propyrium iodide to permeate through the cell wall and bind to it. Okay? So when that happens, we know that bacteria are dead or inactivated. Well, we found a couple of things. These techniques allow us to monitor phenotypic difference. We can also track metabolically active cells. I mean, we can tell the ones that are alive and the ones that are dead before and in the photosensitizer uptake, and so on. These are some of the photosensitizers that we use. They are all known as BODIPI, boron dipyrometrin, bottom the pyrometines, okay, and they generate different type of reactive oxygen species. Usually, type 1, they photosensitize superoxide radical anion and hydroxyl radicals, and type 2, they generate similar oxygen. And these are the bacterial strains that we use in this case, and we have methicillin resistant Staphylococcus aureus, MRSA, coli, Pseudomonas, Klebsiella. What is interesting about all of them is that they are members of the well-known escape pathogens, known for being one of the most, they generate the highest number of nosocomial infections, hospital infections. Okay. So I'm going to show now is a video where what you're seeing here is a bright field image. This grayscale is a bright field image, and I hope that you can see some bacteria. There is a bacterium here, another one here. This is the one that we're going to track the intensity profile and so on. So what you're going to see is how they start lighting up. You have the photosensitizer. The photosensitizer is going to generate damage because it's generating reactive oxygen species, and then pyrium iodide binds to it. So that's the one that we're looking at, and as you can see, this is the density versus time profile. You can see how it's kind of an exponential growth right here. It's kind of sigmoidal actually. And well, and for each one of these, for each bacterium, we obtain these intensity versus time profiles. And I hope that you notice that they light up at different times, right? They don't light up at the same time. Well, for each one of them, we obtain this intensity versus time profile, and then we get some parameters. We define initial intensity. We have the final intensity when they reach the maximum, right? And at this point, we can say that we're confident. We can say they are Hopefully they are dead, they are inactivated, okay? Because all the propylene iodide went in and bind to DNA. And well, and the film clips here are showing that difference, right? The difference in the survival time of each one of the bacterium. So as you can see, some of them live for a very short time period right here, right? When they light up, it means that they are dead, right? So emission means that they are dead, and others, they can live for a very, uh, they live for about 30 minutes, 45 minutes, okay? Because there is a huge difference in this bacterial population, and these are genetically identical to the same isolated colony. Okay. And they are in the same metabolic state. So we are confident and we can say that these are phenotypic differences between between bacteria, right? The different. So with all of those parameters and information, we make a couple of plots, and here I'm only going to show the result for E. coli and Pseudomonas aeruginosa, MRSA. Here, well, this is showing we have final time versus initial time in the x-axis, and then we have some histograms over here that show the initial time and the final time. Same thing over here. And each one of these squares of these dots over here, that represents a bacterium. And well, we found two basic things. The first one is that the amount or the heterogeneity due to phenotypic resistance depends on two things. One is the bacteria that we're using. So I hope that you can remember this is E. coli, is a gram negative, and MRSA, that's a gram positive. So there is usually E. coli has a more resistant level, more okay. So in this case, you can see that if we use the same photosensitizer, here the scatter plot in the case of the Staphylococcus aureus is narrower, right? And however, here for E. coli is a broader scatter plot showing that, well, it takes them longer to die. And in that case, since they are more robust, more resistant to the treatment, then we can clearly see heterogeneity due to those phenotypic characteristics, right? And second is that, of course, it depends on the type of photosensitizer that we are using. So, for example, most bacterial strains, they have an overall negative charge on the surface. Okay, so in this case, if we use a cationic photosensitizer, so it's going to have a better interaction with the bacterial cell wall. And again, we can see how for the cationic photosensitizer, the scatter plot or the distribution is narrow, it's very narrow. And if you look at the histograms here, and then if you compare that to the neutral photosensitizer over here, you can see a broader distribution. Okay. So this is also showing us that with this photodynamic treatments, they need to be optimized to increase the likelihood of achieving this target. Okay, what that probably means that if you have a bacterial infection, and you can see that some of them die super quickly, but rather it takes a long time, right? Or for example, you have a skin infection and you want to use for dynamic inactivation, then you need to make sure that the amount that you irradiate your sample for a very long time period, right? Because otherwise the susceptible bacteria is going to die, and then the resistant bacteria can survive and propagate infection. Okay. So something else that I want to share with you, and this is something that we're doing right now at SIUE. So we use the same kind of the same setup, the chamber that I showed to you before. But now, instead of monitoring phenotypic resistance, what we are doing is monitoring how bacterial motility is affected in the presence of these of antibiotics or photosensitizer as well. Okay? So we are looking at chemiotaxis, and that is the movement of an organism in response to a chemical stimulus. Okay? So how does it work? Well, in the case of bacteria, and this here is showing E. coli, so bacteria can run or tumble, right? If they are swimming in solution, they can run up or tumble by reversing the direction of the flagellar rotation. Okay? So it says here baseline behavior. So for example, you have a bacterium buffer, okay? So they're swimming, they're going to go in a straight line. At some point, they are going to tumble in direction, running in direction, keep running, and so on. In the presence of a chemorepellent, something that is toxic for the cell, they're going to tumble more and run less away in direction away from the toxic, right? The presence of a chemioattractant, they're going to run more and tumble less in the direction of wherever this chemioattractant is, sorry, nutrient, food, something that they like, right? It's the same for us humans, which is go to the direction where we found things that we like, okay? So what we are looking at is how these random walks, that are actually what we are doing here is study how the different levels of loss affect those what are known as random walks, what is known by bibliography as random walks of. Okay? So I just want to share with you a couple of things that we have so far, and this is, in this case, instead of using E. coli, we're using Pseudomonas. And well, we have a bacterium there that is kind of going everywhere. This is kind of in real time, that how they move, how fast they move. And then we use a software to track the trajectory, follow the trajectory of the bacterium. And well, from there you can obtain a lot of here, the amount of directional change, speed, displacement. Okay? So from that, what we have been doing and to make, instead of, we keep using those chambers, but now we have a small agarose pads with a small wells of, as you can see here, like a porous agarose pads. So here we have Imperium iodide, photosensitizer, and these are bacteria that are just swimming everywhere, right? I hope that you can see them or hear how they swim. Then we start exciting the sample. We irradiate the sample with green light, and after 5 minutes, now you can see like there's movement. And after, even after 15 minutes, after 60 minutes, they are not swimming at all. In fact, they are moving, but that's because of diffusion, right? Simple diffusion. They're just, they are floating around, just like toys in a pool. They don't move much, right? So, but as you can see, they are not all of them are emitting fluorescence. They are not light up yet, right? That means that at this point, the amount of oxidants, of active oxidant species that we are generating, well, for sure, they are affecting the chemotactic machinery of the cell, but they are not that yet. Actually, it took us 5 hours to kill them, but at least you see emission from propylium iodide as you can see here is fluorescent. Okay, so some points for this first part of the presentation. Well, we used a novel methodology to characterize oxidative damage in real time. So despite the phenotypic difference between bacteria, no survival were observed after the 3 months done, demonstrating that okay, can take longer, but that's in all the microorganisms showing that they can't survive the 3 months like using antibiotics, right? And then, well, and then a little bit about how this the oxidation species inhibit the bacterial chemical. So the second part, I wanna share with you a couple of stuff that we have topics here to show you now how we use those sensitizer, how we use those to fabricate some antimicrobial materials, and actually these ones with polylactic acid, that's what we are doing right now at SIV. So I'm gonna focus these two over here. And so pretty much what we are doing is use polylactic acid. That's a it's a well-known polymer. It's very commonly used for 3D printing. 3D printing. What we do is we buy pellets, PLA, and then we combine that with different photosensitizers and antibiotics as well. We have this filament maker right here. It's a filament extruder. We make a filament, and we load it into 3D printer, and we whatever we want, and then we test and see the antimicrobial properties of it. If it can kill microbes. Okay. So, for example, here we make different surface, and in this case, we made this device over here. We 3D printed a mesh, and then we sandwich that mesh between the two LED panels. We have water circulating through the mesh, 10 minutes water, and that's how the device works. And I'm going to show a couple of results in the next slides. So, some of the things that we do before start the inactivation is we test the physical properties of the photosensitizers. We want to make sure that they remain the same. We want to see the same photophysical properties of the photosensitizer in solution. And once it is absorbed or absorbed on the material, so as you can see, we make rectangles right here, different loading of the photosensitizer, and then we make these cuvettes or so to be able to measure absorption and fluorescence. And in this case, we can see that, for example, the fluorescence spectrum of the photosensitizer in a zero nitrogen solution is very similar to the fluorescence spectrum of the photosensitizer loaded into or is in the polymeric mat. Okay, so that means that the photophysical properties remain the same. So photosensitizers is very important because if they don't, then they won't generate any results. Okay, so that's why I make sure that they remain the same, or at least that they have some absorption condition. So these are some of the things that we make. A jar over here. All these are similar to chambers that we use our microscope. And then, as you can see, for cover lid right there, then have a thin coating. We 3D printed a thin layer of the polymer on top of the cover lid because we want to see in real time how long it takes the material to inactivate to kill microbes, right? That's with the idea of using this in real world applications. As you can see, we made some door handles. So we want to know how long it takes the material to kill the microbes. And for that, we just use our microscope and iodide, just like we did before, to see in real time how long it will take. In this case over here, we just use a Q-tip and we pick up, lift up bacteria, and then that goes into an agar plate, and we keep it over. And compare that to a regular normal door handle, for example. Well, and this is the device there. Of course, all of this material needs to be exposed to light, either room light or in this case sunlight. Otherwise, the photosensitizer is not going to generate cytotoxic compounds. So here are a couple of results, and this comes from the Max. And we thought, well, maybe you go camping and you need some clean water from the river or whatever, and then you can just use one of these jars. Well, the jar is very small; it's only 100 milliliters. So, and it took like more than about 6 or 8 hours to clean those 100 milliliters. So I'm gonna save you, but yeah, because maybe we need a bigger jar. And then these are the two bacterial strains that we use, gram positive and a gram negative. And well, after those 8 hours, we found a complete bacterial inactivation or elimination for either the gram negative or the gram. Okay. In this case, this is scanning electron microscopy, and now this is showing here we have native PLAs, this PLA that haven't been modified, and we grew a biofilm. Okay. This is a first stage of a biofilm formation. Over here we have the antimicrobial material in the dark, so you can still see some remains of the biofilm over here. And then this is the antimicrobial material plus light, and we found a complete biofilm eradication. Okay, after the treatment, of course, after exposing each one of these materials with light. So, and well, and in the case of this device over here to clean water, actually it is a mesh, but it's not a filter. It's a mesh to increase, to improve the amount of oxygen that is being generated. So it's kind of, we are increasing the surface area of the material, that's all. Okay, but it's not a filter, it doesn't filtrate, it only increases the surface area because if the bacterium is not close enough to the surface, then it won't react with those reactive oxygen. That's why we have that mesh. And well, in this case, this is showing how the mesh works, that actually showing that reactive oxygen species are being generated. Okay, here we have a singlet oxygen trap. Okay, so singlet oxygen is a reactive oxygen species, and we use this molecule, and we just monitor the decrease in absorption, and that shows that singlet oxygen is being generated. Same here, this is emission of singlet oxygen in the near IR. And that peak right there, that's for our material with the photosensitizer. Well, and actually this is the plot that we care about, the data that we care about. And before irradiation, there was no solution or water decontamination, as you can see here. And after 60 minutes of exciting the mesh with light, we achieved 4 logs reductions in the colony. Actually, about 99.9 of bacterial. It was very good. So these materials are stable, they can be reused, we can recycle them. Well, we can recycle them, but sometimes we need to add a little bit more of photosensitizer because after recycle them a couple of times, the photosensitizer will degrade, thermal degradation. It works anyways, and recycling is very good. So summary of this second part, well, we show the photosensitizer attached to different they're going to generate reactive oxygen species, and then the materials have great potential in the field of cell therapizing material. Okay, and finally, okay,
It's 4:10. We have time.
okay, it's almost done anyway. So. And finally, well, now we learned that actually a lot of these reactive oxygen species are generated in the environment and by natural particulate matter. So then this led us to study some oxidation process in the environment, but at the aerosolic index. Okay? What I mean with that is that in a previous work, we made this combination of particles. So one of these is the photosensitizing particle, okay? And the other one is the trapping particle. The photosensitizing particle, of course, and then the trapping particle have different reactive ROS quenchers. In this case, singlet oxygen quenchers. But now everything happens in the air. right? We are not using any solution. But well, actually, air is a solution. But I mean, we're not using any liquids. Okay, it's just gas, pollution, air. Okay, and that single oxygen, that reactive oxygen species, is airborne. Okay, so it's gonna fly from one particle to the other. You can see right here. And so, for example, the solution, in solution, the diffusion distance of ROS is only a couple of nanometers, but then in the air, it can diffuse up to at least 10th of millimeters. So it's at least 5,000 times. It can go 5,000 times farther. So in this study, we were monitoring the emission of singlet oxygen using expensive equipment and so on. And that gave us a lot of information about the oxygen lifetime. But it didn't give us a lot of information about how far that single oxygen can go, right? And also about the lifetime of the aromatic compounds that we have here to trap single oxygen. So now what we are doing is to use, of course, a microscopy. And what we did is we have a particle right here. And now this is loaded with zinc phthalocyanine, a different photosensitizer. And we have another particle over here, and that's loaded with a furan, okay, a trapping molecule, a singlet oxygen quencher. And these particles are made of silicon dioxide, sand, green sand. That's all they are. And we use sand because it's one of the main components in soil. 30 depends on the region, but 30 to 50% of soils is silicon dioxide. Okay. But what we're doing, if we have the sensitizing or sensitizing particles over here, the trapping particles over there, and once single oxygen flies or is airborne from here to there, this trapping agent is going to quench or trap singlet oxygen, okay? And it's gonna get oxidized. Okay, so when the molecule is oxidized, it goes from a fluorescence on state to a fluorescence off state. And this is what we monitor, the decrease in the fluorescence intensity. Okay. So again, making more of our devices and chambers. And here we have a chamber. So we mix the particles. We put the photosensitizing and the trapping particles. And then we purge everything with oxygen to increase the production of singlet oxygen. Okay, we increase the concentration. And then, what happens is that the quantum fluorescence quantum yield of the photosensitizer is very low because once we have the molecule in the excited state, everything goes to the triplet side. So the fluorescent quantum yield is super low, okay? And so we can use the fluorescence intensity to then find which ones are the photosensitizer particles and which ones are the trapping. Okay, so the ones highlighted or that have that green contour around, those are the trapping particles. Okay, and you can see them here, very bright, right? And then the other ones over there in orange, those are the photosensitizer particles. So we monitor the decreasing intensity of these trapping particles, and we can obtain different information. For example, the lifetime of that furan molecule or the furan molecule, and so this right here is showing control, just quencher particles over here in blue squares. Then we have particles of silicon dioxide, no photosensitizer, plus the quencher, that's the black squares. And finally, we have the mixture of the sensitizing photosensitizing particles plus the quencher right there. So you can see the absorption. Okay. So and we also found that our technique is very sensitive, and the goal of all of these we're using a furan, the molecule that I just showed, and that furan per se is not an organic pollutant, but it serves as a model for other organic pollutants that are involved in a lot of different diseases. Okay, those furans derivatives are generated, for example, during waste incineration and so on. So they're everywhere in the environment. So this will give us another way to study degradation of those organic pollutants. And what this is showing here is the sensitivity of our technique. So we prepared particles with different loading of the trapping molecules of that furan. So you can see that we can detect our this is the lowest emission. This is our detection limit actually. Okay, using a 10x objective. You can see that for the lowest loading of the trap, if we increase the objective magnification, we can't even get our intensity back, okay, because 100x objective collects more light, that's all. But if we look at this one over here, we found that we can detect pollutant in a concentration as low as 1.4 picograms per gram of particle, okay, with this loading over here. Okay, so and finally, something else that we have going on is to see what we want to see is how this airborne singlet oxygen, this ROS, reactive oxygen species, can affect if it does, if it have any effect on the soil microbiome and endolith microbes that live in rocks, within rocks and so on. And well, a couple of conclusions from this last section over here. Well, we found a way to easily detect and monitor experience. We have been developing strategies to monitor in real time the production of singlet oxygens in different environments. And what we plan to, or we are trying to think how different oxygens affect the soil microbiome in air solid ingredients, right? Again, not using any liquids. So with this, I hope that you enjoy my presentation, where I share with you how we explore the oxygen species, those photosensitizers, those photoactive biocides, effectivity, oxidative damage, activating materials, and then a little bit related or to the environment, trying to study some environmental processes. I would like to thank my students, current students, people working in my lab, and of course, over here, former students as well, my collaborators, to all of you, to Maryville University, and to SIUE for all of their support. And of course, I will be happy to answer any of the questions. And this is something else that we're doing now, but I think so. Thank you very much. Okay.
Thank you for your presentation. I just had a quick kind of clarifying question. So in the beginning, you talked about the photosensitive molecules and monitoring how they created the reactive oxygen species. Is that correct? And then did they also cause those microbes to fluoresce, or was that another dye?
That's another.
Okay,
Yeah.
so there's like two different things.
Yeah, we need to use two different dyes. So for sensitizer, they usually the fluorescence intensity is super low. Everything goes to generating the energy that they absorb goes into it. And sometimes they don't. They might not bind to the cell surface, or we can send
activated materials such as
Well, actually, I would say that the biggest challenge was to make the filaments. But other than that, well, we need to do a lot of optimization, a lot of math before making. So, we calculate the difference for the sensitizers in the material because if they actually close, they will self-quench. So instead of generating reactive oxygens, they use that energy. They transfer that energy to another molecule. So we need to with that, we need to be careful in the sense that the photosensitizers that we are using is not affected when it's treated in the polymeric matrix. And that will be the biggest challenge.
Okay, so you were talking about the genotypes, right? So I was wondering which one to be worse.
We don't, that's as far as we go with phenotype. We know that is because of phenotype. And when I was doing research at the university, what we try to do is to see, yeah, well, we're saying that it's the phenotype, but we want to show the single cell level, which are the difference or the phenotypic difference between each bacterium. But it's super hard because we're talking about per time 2 micrometers of microbes, right? So we try to instant cryo cryo-EM, electron microscopy, and that was coupled to an IR to see if we can follow and see some of the lipids that have been oxidized between bacteria, right? So we found that some of them have a higher signal or lower signal, but it's too small.
Yes, hello. I was wondering about what solar panels the potential solar panel for like the solar bacterial filtration that you were talking about.
Using solar panels. So, well, we can use solar panels. That's actually a great idea. But instead of we need to use those solar panels to generate light, right? And then that light excite the photosensor. But that's yeah. Thanks. You mind if we can just add a solar panel to our device and but if you want to come visit it out. Okay.
Did you use green light because it's the most efficient debate for the synthesizer, or was there another reason?
So, well, it depends on what you want to do. Usually, we use green light in this case because that photosensitizer absorbs the green region. But for example, for material is great, it is great. But in cancer research, they use this for dynamic therapy. They have to use light. They work in what is known as the therapeutical window. It's a region where it doesn't absorb any light. So we use very much.
So you mentioned at the end how you're studying the oxidants and the effect that they have on the soil microbiome, especially at that soil-air interface. I was just curious if there's applications for like modern agriculture and making our agriculture like systems more sustainable and efficient through that technology.
Yeah, there is huge field. A lot of people is working on that, especially using those photosensitizers to eliminate different plays, microbes, and so on. I don't do any of that because I just feel that it's not my field. But I mean, if you want, I can send you papers. It's a lot of important.
So this is more question. You mentioned that it's formula infections is safe. Is the photodynamic activation
So that therapy, as far as we know, we can kill all of those microbes, including virus and fungi. So you can kill all of those microbes because they just generate so much oxygen species that the defense mechanisms just can't eliminate all of those oxygens. So far, there are no reports showing that bacteria or other microbes have developed resistance towards this kind of thing.
So when you were talking about making objects out of the filament to be antimicrobial, you were talking about like reusing them and recycling them. I was just wondering, is the window where they lose efficiency, is it like long enough to be profitable and useful in the real world, or is it shorter?
If you want to recycle them, I mean, the lifetime of these materials is super. I mean, we still have them in the lab. Once the photosensitizer is embedded in the polymeric matrix, it's very well protected. And I don't know, I have some of these materials when I moved from somewhere else to SIUE over in 1.5 years that we just left them in a solution and you can still see the dye. They're very stable. But if you want to recycle them, for example, the mags that we made, right? If you want to recycle that, well, what can degrade that photosensitizer is the high temperature that we need to use in the extruder, in the filament maker machine. That degrades some of the photosensitizer, at least 10%. But other than that, they last a relatively long time.
So going back to part one, or part A, where you talk about using reactive oxidative species as an antimicrobial, was that like for medical purposes, or is that just like general lab use?
Well, that's what we would like to know. And what we're trying to find is one kind of connection. This phenotypic resistance and real world infections. We're trying to find that connection here. And that's why now we're also working on the chemical project, because based on those phenotypes, some bacteria, they're going to swim more or just move faster, do something different. Right? So we want to know how far they can go, how fast they can go, just infect more tissue.
So, are there experts like if the actual diagnostic species becomes something like whether or not that will like impact
Well, it doesn't. It has been used. It depends. So FDA approved a lot of. There are a lot of photosensitizer approved by the FDA, especially some porphyrins and rose bengal, some of those. For infections, is fine because usually to kill eukaryotic cells, you need to the treatments. It takes longer to kill eukaryotic cells. It takes 3, 4, 5 hours, depending on the process. To kill bacteria, it can take like 30 minutes. Okay? And also the way that those photosensitizers are uptaken or bind to eukaryotic cells or prokaryotic cells is different. So the positive cationic photosensitizer will bind. You're going to have a high concentration of the photosensitizer on the surface of a bacterium, but it's not going to bind that well. So you can kind of play with those conditions. Or say.
Are the treatments you're speaking of carcinogenic, potentially? Like cancer causing?
No, I mean, of course, they can generate mutations because we're generating active oxygen species. They can oxidize your DNA, but not at that level.
Oh, right. Thank you so much. I want to be mindful
you
of
very much.
everyone's time. So everybody else can go, but if my advanced topic students can stay behind for a little bit, that'll be awesome. So Thank you so much for coming.